So welcome everybody to the next iteration of the IITE seminar series. It is my great pleasure to welcome Lauren Hallett this week, who will be talking about integrating population and community synchrony across scales. So without further ado, please, Lauren, the floor is yours.
Well, thank you so much, Jerry. And it's really a pleasure to be here. I've been really enjoying catching up on all the YouTube videos of past seminars. This is an awesome series, and so it's an honor to participate. And so today I'm going to be talking about some work from a working group I led a few years back on integrating population and community synchrony across scales. And to kind of just kind of situate you with what the task was that we had before us. There's really two bodies of literature that have used this term synchrony and thought about concepts of synchrony. And the first is thinking about spatial synchrony, and this is also known as population synchrony. And this is where you have different populations of the same species across a landscape. And so, in this case, we have got 4 patches where that species is fluctuating over time. And it's quite common that you can have populations that are disparate that end up cycling in synchronous ways. And so what that means is that the landscape scale, you can have strong overall fluctuations in the species because of synchronicity between patches. And this has been thought a lot about in terms of pest outbreaks. And so if we're thinking about trying to plan for pest outbreaks, understanding synchrony is really important for seeing when the landscape might be really heavily hit by pests. And it's also been really important from conservation. So if you have kind of this landscape level synchrony in a species of conservation concern, then if the population undergoes a period of time where it is low across the entire landscape, you have a higher risk of stochastic extinction. So there's been a big body of literature on spatial synchrony. Over in my world as a community ecologist, I've thought a lot more about community synchrony. And so in this case, instead of having multiple patches, we have just one patch. We have multiple species. And the idea here is if you have synchrony between different species within that patch, then the overall aggregate abundance of all species, and so the production of the system or the cover of the system, will be highly variable over time. And so from my background as a community ecologist working in rangelands, I think about this a lot from a forage production perspective. And what happens if all the species are fluctuating synchronously? You get hit by a bad drought, then you really have nothing there to compensate. And so I was interested in this, and then I started talking to some population synchrony folks, particularly Dan Ruman's group, and we were like, we should really integrate these approaches and questions with population and community synchrony. And what do we have these different disciplines to learn from one another, both in terms of methodologies and also in terms of concepts. And so to do that, we brought together the LTER Synchrony Working Group. So this was funded by the LTER office, but hosted at NCES, or the National Center for Ecological Analysis and Synthesis. And so we brought together groups of theoretical ecologists, empirical, field ecologists, and people from the community synchrony and the population synchrony side of things. We said, let's really think about this. And so I'm going to present some work from this group today. And this is a gold star cast. Everyone's wonderful, but special gold stars are going to go to Lei Zhao, who led the first paper that I'll be talking about. Lauren Shoemaker, who do a lot of collaborations with. So last week, I think she talked about some of our collaborations, and this week I'll jump more into some of the ones with her. And she also downloaded some slides for this. And then John Walter, and he's going to cover some of the spatial synchrony, bringing spatial synchrony in, and also donated some slides. So when we think about what even is synchrony and how do we measure it, one of the things as a community synchrony sort of minded person that really valued from the population synchrony was, okay, so if we're thinking about fluctuations in populations that are correlated through time across multiple locations, you might have all these different fluctuating populations. But if they become synchronous, like how do we measure that? What are the population synchrony people doing to measure that? And one of the things that's really big in population or spatial synchrony is that fluctuations can occur at different frequencies. And so they often adopt a wavelet approach to synchrony, where they say, okay, there might be drivers at kind of larger time scales like climate that are influencing synchrony. And so we would see kind of a long time scale fluctuations. But there also might be dynamics that are happening at short time scales. And so the interplay of this is best teased apart from kind of a wavelet or spectral approach. And just to give an example of some of the types of work from this is a paper I think is so cool by Lawrence Shepard and colleagues, as they found that when you think about spatial synchrony at a timescales perspective, you can see how and when it shifts from long to short timescales. So to walk you through this diagram, this is a wavelet mean field. On the x-axis here, we have the time scale of a fluctuation. So over here at 2 would be the highest frequency you would have, up 1 year, down the next, up 1 year, down the next. Over here at 20, it'd be like a 20-year decadal or multi-decadal oscillation. So this is the time scale. This here is their time series. And so this is a record of aphid outbreaks from the 80s up to 2010. And then the heat map is showing the level of synchrony. And so here we see that early in the time series, so from the 80s and the 90s, we had a lot of synchrony in aphid populations that were occurring at a decadal scale. But it shifted, and in the late 90s and in the 2000s, most of the synchrony in populations occurred at short time scales. So that was really interesting. If you're trying to find a signal of synchrony, this time scale effect can matter. But another really important point of this is that if you look at the timescale of synchrony, you can also start to see how it relates to a timescale of drivers. And so winter temperature is really important in controlling aphid populations. And what Lawrence and his colleagues found was that there used to be kind of a much more decadal signature of synchrony in winter temperature. But as we're progressing through time, we're actually seeing that occurring at much higher frequencies. And so that's driving this shift in aphid patterns. So I think this is a really important lesson from spatial synchrony. And so let's put a pin in that. And now let's think about what's happening over here in the community synchrony world. And so over in community synchrony, for a long time, we've really focused on community synchrony as it's linked to stability. And so this idea that if species are fluctuating in tandem in the same patch, you're going to have really variable ecosystem function. And conversely, if they are fluctuating out of sync, so if they're trading off over time, you're going to have what's often termed compensatory dynamics, but greater stability, because one species compensates for another as it rises and falls. And a very classic way to measure this has been the variance ratio. And so what the variance ratio does is it compares the variance of the whole aggregate community to the variance of every individual population. And when you do that, you basically are aggregating the whole time series, and then you're getting this single metric for the time series where greater than 1 would suggest synchrony, right? So the overall variance of the community is really high relative to individual populations. Less than 1 would suggest compensation. And so the variance of the community is low relative to individual populations. And this is just one of the community synchrony metric tools. There's a bunch of others that look at synchrony. There's some developed by Michel Leroux, some by Nicholas Gross, but all of them tend to aggregate across the whole time series. I think the variance ratio is very useful. So some of my earlier work, I used it to relate synchrony and stability across environmental gradients. And so we were like, what would be driving, increase in compensatory dynamics? And the first thing we found when we looked across a lot of grassland sites is that community stability indeed does increase with these sorts of trade-offs. So over here with a low variance ratio, we see very high community stability. When the variance ratio is high, species tend to be more in sync. And we thought maybe one of the things that could be happening here is if you have a variable environment, it might be selecting for different species over time. And so we thought, okay, what's driving this change in the variance ratio? We thought a lot of these grasslands are driven by precipitation. So maybe, and indeed we found that there would be more species trade-offs or a lower variance ratio. There's really high precipitation variability, like a wet year would favor some species, a dry year another. So it's cool, it's a nice metric, and it allowed us to make some inference across environmental gradients. But there's limitations to it too. And just to highlight that, I'm going to hone in on one of my favorite sites. This is Jasper Ridge. It's got super high precipitation variability and also a lot of species trade-offs. So if we think about Jasper Ridge, so here's a plot of what Jasper Ridge looked like back in the 80s. So it's a serpentine grassland. It's got a lot of these really small native forbs. And if you kind of look at this snapshot, it's, okay, there's a lot of Lithia, California goldfields, happening here. But my colleague Richard Hobbs and now I have measured this site every year for 40 years now, as of this April. And one of the things that we see is it's not just a classic, oh, it's Lithia and then another species and then back to Lithia. The whole system is bopping around from one kind of dominance to another. And so when we take a single value like a variance ratio to represent this whole time series, we obscure a lot of the pattern and process that is governing these sorts of trade-offs or synchrony in species in the landscape. So working group, we thought, how can we learn from some of this timescale specificity that's happening with the population synchrony and bring it to understand what's going on at a community level? So we thought, what are some of the drivers that are going on at this site? And one of them clearly is the rainfall variability, right? It's at the extreme end of all of our grassland sites in terms of just the degree of variability. And to highlight it, this is the time series. We had some years where we only had like 200 millimeters of rain. And then some years where we're almost at 1,200 millimeters of rain. I mean, that's insane variation, right? So that is clearly a signature of the system. But the second is that this is a landscape that has a lot of gopher disturbance. And so Richard Hobbs's this really seminal work showing how disturbance supports diversity was in the system. And we said that there's also a lot of variability in gopher disturbance over time. And then finally, we have species interactions. And so the species that are in the system have different life histories, they have different competitive abilities. And so if they're responding to these different drivers in different ways, that could yield some more complex patterns. So we thought, okay, can we take this lesson from population synchrony to community synchrony? And the way we did it is that Lazout developed this very nice timescale specific variance ratio. And so what he did, and this is with Dan Ruman and Xiaopeng Wang as well, is instead of just taking the variance of a time series, we replaced it with a power spectra. And then we replaced a covariance with co-spectra. And that allowed us to basically decompose the variance ratio at any level of frequency and see where you had a higher or lower variance ratio metric at a given frequency. And so Emily's got a nice paper in Ecosphere, and then also we put an R package together, TSVR. So we said, okay, let's apply this timescale specific approach and see if it improves our understanding of community dynamics. And in particular, we thought, can a timescale specific variance ratio help us disentangle environmental drivers operating at different timescales? And this is where Lauren Shoemaker comes in. She does really great modeling. I do empirical work. So we said, okay, let's do a model and then we'll test it with this empirical system of Jasper Ridge. And so as a really simple kind of proof of concept example, imagine that we have, one driver over time that's at, relatively, a high frequency driver. And maybe we have two species, so gray and black, and they are both in sync with this driver, leading to greater fluctuations in total biomass. But maybe there's a long time scale driver. And so, maybe we've got like the Pacific decadal oscillation going on, and maybe there's a compensatory response between these two species to the long-term driver versus to the short-term driver. If we put that as a composite, what we're going to see in these time series is we're going to see patterns of synchrony over long time scales, or synchrony over short time scales and compensation over long. But actually, if you were to try to just look at a correlation, it's going to be really hard to see what the correlation is because of these different time scale drivers. So we said, okay, let's take that composite, and now let's see, is our time scale specific variance ratio able to parse it? And indeed, ta-da, it did. And so what we can see is when we look at short time scales, so we said basically, 4 years or fewer for fluctuations, we were able to pull out that we had really high synchrony occurring at short time scales and a lot of compensation at long time scales. And so that worked. And another thing that we found was, kind of in a lot of simulations, actually all of them, the classic variance ratio was really biased by these long time scales. So that was kind of overriding these short time scale drivers, which it's interesting. And so there's, kind of put a pin in that for what that would mean for a lot of our studies that they're often shorter term. And so, when we think about this, a classic variance ratio would miss a lot of those drivers that might be important. So we thought, okay, this is a nice thought experiment. But let's think about it empirically. And, two species that I thought, this is probably really relevant for is a Microceros douglasi and Plantago erecta. And Plantago is the dominant form in the system. It tends to be really drought tolerant. Microceros seems a little bit more acquisitive, maybe likes more water, but nobody likes to get decimated by a gopher, right? And so at short time scales, if a gopher comes and is like a mini bomb in your system, that's Dan Ruman's term for it, then everyone's going to go down. But at long time scales, maybe they have a very different response to climate. See, in this time series, we chose, okay, we're going to start the record when there's a gopher disturbance, and then pull from the record periods of time that then follow that after. And what we found was really kind of following our expectation that we had a much higher variance ratio or much more synchrony at these short time scales, which you would expect if you're being driven by gopher disturbance at short time scales, right? You get bombed and then seeds come back in and you rebound relatively quickly. Versus at long time scales, we had a much lower variance ratio, likely we would think because of these kind of different responses to long-term climate patterns. And again, our classic variance ratio was really biased towards that. And so when we did this cross-site synthesis, the patterns we're seeing in that cross-site synthesis are driven by these long time scale dynamics. It's pretty cool. But then there's more you can do with this. So you can start to say, okay, that's one mechanism of different environmental drivers. But you also might have a lot of endogenous dynamics that are operating at different time scales that could cause you to have these patterns. Even if species respond, directionally to the environment in the same way. And kind of as an example of that, we thought, okay, let's think about a situation where you have, say, 3 different species, and they all have a directional, shared directional response to a driver, but they respond at different rates. And so here we have our black species, and it responds, kind of just tracks the environment. Our pink species responds really rapidly, so it responds quickly, then tends to overshoot. And then our color of gold species has a lagged response, and so has a slower response to the drivers. And so in this case, we're going to say they both have the same, all the species have the same directional response. And if you have 2 species that have the same time scale of response to, then what you see, of course, is a lot of synchrony. So in this case, both species, this black and this dashed black, can trace the environment completely. And so there's 2 drivers, short and long time scale, that are at this, that they're both responding to, and you have a very high variance ratio because they're both tracking all those fluctuations. So they're completely in sync. But if you have species with different timescales of responses, what you'll find is that even if they're responding directionally the same way, you're going to end up having more compensation than you maybe would expect because of this lag. And so this is pointed out by a paper by Michel Leroux and Claire de Mazencourt in 2008. And what this timescale specific variance ratio means is we can really parse this effect, where what we see is at short time scales, there's some disturbance, and they respond relatively in tandem, but at longer time scales, a lag in the response is going to end up putting them offset the response so that they peak at different times, creating more compensation. And again, kind of interestingly, this phenomenon is stronger at long time scales than short, leading towards an ecological bias towards long time scales informing the variance ratio as well. Which I think when you start to kind of put the initial example of maybe different directional responses to environmental drivers together with this life history and how life history is going to shape dynamics, it's often not so simple as species A likes one environment, species B likes another. It's, do species A and B like the same environment when it's rain versus gophers, and do they respond at the same rate? thought, okay, if we're at Jasper Ridge, how would this play out? And one thing we thought was, okay, we have mostly annual species. So here's our Plantago, that dominant annual. But there are some perennial grasses. So here's Elymus glaucus, this nice native perennial grass. And we thought, if an annual species gets bombed by a gopher, probably there's a pretty good seed source just outside of that gopher disturbance, so it can recover pretty quickly. Versus a perennial grass that gets bombed, it's probably going to be a lag where it needs to be kind of reoccurring its rootstock if it didn't get killed completely, or it's going to be much slower if it has to recruit from seed than an annual. And so we have a scenario here where we did a similar thing. We said, okay, we're taking all the time series where they get gopher bombed in the second year, and then we're going to kind of look at how this plays out in terms of their variance ratio. And indeed, what we found that this pattern is a little bit more muted, but it's directionally as we would expect. That again, at short time scales, there's synchronicity. Nobody likes the gopher bombing, but at longer time scales, we have a little bit more, a little bit lower variance ratio, because even though these are two species that we think have pretty similar climate responses, their ability to recover is going to be driven by these different timescales, and so we see this kind of trade-off, or not. We see the pattern of trade-offs, even if they're responding directionally in the same way. Finally, because Lauren likes to think more spatially than me, and it's great because it's dragging me into that world. She was like, listen, those patches are not in isolation. You've been talking about the annuals having dispersal into them as a rapid way to respond for a long time now. Let's think about what dispersal would do if you have patches with different environmental responses. And so in this case, we have patch 1 happening in the first row, and patch 1 in this model example is responding to a short timescale driver, and in this case, they're responding in sync. Versus over here in patch 2, there's a longer timescale that's really affecting it, and in this case, they're trading off. And we thought, you might see differences in timescale of response to environmental drivers. If your soil type is shallower versus deeper, it changes your water holding capacity. Maybe the amount that you're really tracking, rainfall fluctuations could be changing. So maybe one reason for a landscape difference in patches. In a world with no dispersal, we see really what we would expect, which is at patch 1, when they're very synchronous with the short timescale driver, it's a very high variance ratio. Patch 2, that's very compensatory, has a very low variance ratio. And at a landscape scale, there's short frequency, there's a high variance ratio at short drivers, low variance ratio at long, and it kind of averages to a lower classic. But if you add dispersal in and you get propagules going back and forth between patches, what it does is it shifts the dynamics such that you end up seeing the signature of the environmental variation that your neighbor felt. And so over here in patch 1, we see that we now go from having a classic variation ratio that's very high to it being somewhat lowered because of propagules coming from the patch that is governed by the long timescale driver. Conversely, for the patch that is governed by the long timescale driver, having propagules coming in from that short timescale patch means that we see some of that signature, even though that driver isn't in the patch, is being driven by dispersal, with a net fairly similar landscape effect. So we weren't clever enough to really think about how to do this empirically. We thought it was pretty important, like at Jasper Ridge per se. We thought it's really kind of an important point where when we're thinking about these drivers of community synchrony, it's, it can be driven by multiple environmental drivers, can be driven by different life stages, and also by who your neighbor is. And all of that aggregates to really complex dynamics that a single metric of one variance ratio over time is going to obscure and miss a lot. So then we're like, okay, let's go bigger. Let's think about this across sites, and how typical is Jasper Ridge across these sites. So we took the same time series that we'd done that earlier paper, and what Leigh found was that Jasper Ridge is really, has this very strong signature of high synchrony at short time scales, very low synchrony at long time scales. There's some other sites that have that pattern, and then there's some sites like the Sevietta that it's really not having this time scale specific signature. And we thought this is interesting and worth further exploration. Jasper Ridge, we thought this short, high, frequency or high synchrony at short timescales, this site is really driven by gophers in a way that some of the others aren't. Versus over at Savietta, where it's a desert site, maybe everything is being in sync with rainfall, and so there's one really dominant driver, and so you don't get this partitioning. Cool. All right, so let's keep going with our Jasper Ridge example, but now let's do a recap, and then we'll keep going with Jasper Ridge. So in terms of how timescale specificity enhances our understanding of community dynamics, the first is just to really bear in mind that the classic variance ratio is biased towards long-term drivers. And I think what that means is when you do an experiment to test the drivers, for example, my dissertation, I looked at these long timescale patterns across grassland sites, and then I did a 3-year experiment. And maybe that wasn't enough because the drivers that were really governing these patterns were occurring at longer timescales. Multiple drivers are often operating, and it's important to try to parse that, and the timescale specific approach really helps you do that. And so by using this kind of new tool, we could get more insight into the ecology of the system. Okay. The second kind of lesson for community synchrony from population, and this is going to be a bit more of a vignette just to kind of get you thinking about the directions we were headed, is that spatial synchrony thinks about spatial patterns, and community synchrony hasn't as much. And there's been a nice push. So Xiaopeng Wang has these nice papers where it's really looking at patch trade-offs and not just species trade-offs. And so we've there's been a body of work that's thought about it from that perspective, and it's really cool stuff. But we thought, could we also use some of these tools from spatial synchrony? So this is a paper that John Walter led a little while ago that's looking at spongy moth outbreaks. And what they found is that, yes, outbreaks vary spatially, but then the geographic patterns vary by time scale. So this is really cool. So at short time scales, we see this kind of level of connectivity between patches, and there's kind of a lot of trade-offs, versus at long time scales, so lower synchrony, versus at long time scales, there's a different series of patches that are connected, many more patches that are connected, and much higher synchrony. And so, but this is really cool. There's this spatial pattern to the temporal partitioning. And John's term for this is essentially it's geography of synchrony, that some patches are connected at some time scales, some patches at others, and trying to understand that can really help you understand the landscape. And I think when we look at Jasper Ridge, just as we thought, man, trying to condense this whole time series down to one single variance ratio misses a lot. A single value representing the whole space will obscure a lot. So even if we just take the variance ratio in each replicate and then try to, take the average, that's missing a lot of the pattern and process. And when we think about this landscape, one of the main things that's governing it is that you have this spatial heterogeneity. So here we have these rock outcrops, it's really shallow soils. Over here, it's a little bit deeper, and this is happening over the course of just meters. And then again, we've got our gopher that's just bombing, the site, and kind of not entirely random, but sometimes random areas. It's often correlated with deeper soils too. So we thought, can we understand these landscape patterns, and to just kind of put the same ideas in a different way, we can think about this as kind of the coherence between species. So how much they're in lockstep, and the phase. And so in this example, they're very synchronous, and so they're very coherent together, and they're also in phase. So we get this kind of classic synchrony. The compensation would be out of phase, but again, they're very coherent, they're in lockstep, but they're out of phase. You have a quarter phase, which we saw with that the annual response versus the perennial response to the same driver. And then you could have a period where they're not coherent. And so this is kind of another way of kind of quantifying the synchrony, is looking at this degree of coherence in the relationship and then the phase. And so we thought, okay, can we add the spatial specificity to enhance our understanding of community dynamics? And one thing that John has developed is an approach where you basically look at these spatial synchrony networks. to disentangle drivers. And then you can combine it with doing a matrix regression to test the relative importance of those drivers. And what we saw, and this is just a kind of a teaser of results because it's a complicated landscape, but that two of the really dominant annual species, so Plantago and Lysthenia, these anti-phase relationships with gophers, which kind of makes sense, right? The gopher bombs you, your population goes down. The gopher isn't there and you're able to rebound. And so that makes sense. But other species like Vulpia didn't. And with that, it's kind of interesting. Vulpia is one of the few annual grasses in the system. It's a little bit taller. Maybe it recolonizes so quickly that you're really not seeing these types of patterns because its seeds are taller, it can get to those patches. But we also saw that we had patterns between the species that were often unique to between the species. And so Plantago is one of the dominant species in this system that it might be kind of competitive, have a competitive advantage. And indeed, we saw that there was this quarter phase relationship where Vulpia was often lagging Plantago. So Plantago went down, that gave Vulpia an opportunity to increase because that didn't have that kind of competitive effect there. Versus Lysthenia and Vulpia were often in phase with one another. And so maybe they're responding to similar environmental, like climate gradient, or maybe the degree of competition, like that release from Plantago was benefiting both. And so kind of by doing this and parsing different degrees of phase relationships between environmental drivers like gophers, and then between different species, we could see these site level relationships starting to emerge. And then we could look at it in that kind of synchrony network, similar to the spongy moth. And so in this example, we've got these different nodes, they're corresponding to plot locations. And then we've got links between different plots for the degree of spatial synchrony between pairs. And what we found was that the spatial structures are pretty dissimilar between these different species. And so when you have an aggregate of, okay, we've got some site level drivers like rainfall, but we also have these patchy gopher disturbances that species respond to in somewhat different ways. And then we layer on top of that the competitive effect of one species to another and how that can be in kind of quarter phase relationships. You get these very kind of spatially structured synchrony networks per species that aren't always structured in the same way. So I think this is pretty cool. And I think some of our lessons from this are, one, that a geography of synchrony can occur over small areas. And so a lot of this work, like pest outbreaks across the Northeast, we've done really large areas. And here we're seeing that you get this kind of partitioning from different, combination of dispersal and soil and gopher disturbance that's happening at really fine scales. It's in the system of small plants. Second, that drivers at different spatial scales can create these complex landscapes. So you can get these strong relationships between a driver and a response variable, but because there's a spatial partitioning to that driver, you get this really heterogeneous landscape. And that finally, by having that spatial complexity, you can increase landscape stability because different things are doing different things in different places at different times. So species are having this kind of this network of responses that differs from one to the next. All right, so that was kind of a recap through kind of the lessons from the LTER synchrony group. And I just want to make a final plug for working groups. They're awesome. And so I've learned so much by interacting with people that just approach the world, slightly differently than me. I mean, we're all kind of ecologists, but we come from it with different, biases, different perspectives, and it's made for a group that, has been pretty productive in terms of publications. So I'd recommend Xiaopeng's got a paper I didn't talk about that looks about spatial hierarchies, and we have another paper that's looking at synchrony of community properties. But these are the ones I highlighted today. But then also, the group got really interested in making tools. So if any of this appealed to you, there's tools to do it. And so the TSVR packages on CRAN, it does that timescale specific variance ratio. The WSYN does that wavelet approach, similar to the example I gave of Lawrence's paper at the top. And then MMS does matrix model selection for doing that kind of geography of synchrony approach and really looking at drivers. And then finally, if you want to expand on any of these sites that we talked about, or you want to audit our work, the the data packages are available too. And so if you're interested in long-term dynamics, I think they're really usable packages to then use to test your own questions. So with that in mind, I want to acknowledge again the Synchrony Working Group, especially Lei, Lauren, and John, as well as Richard Hobbs, who collected 30-something years of Jasper Ridge data before he passed the baton to me, and NCs, and then the Long-Term Ecological Research Network for putting us up and funding us. And with that, thank you guys so much.
Thank you very much, Lauren. As usual, if you have questions for Lauren, please raise your hand in the participant list and I will call on you. Any questions? Well, people are thinking, let me ask a question then, which is just about the data that where, in what format will this be published? That's amazing, by the way, that you're publishing it.
Oh, yeah, they're on the Environmental Data Initiative. So they're, did I give the citation for it? Oh no. No, I didn't give the citation, but I can post it around. You can link it with the video, but the EDI has been an area where a lot of the LTER data sets are moving towards, and so we published it on there. So highly recommend them in general, and then if you want our packages, the Environmental Data Initiative.
Thank you. In the meantime, oh yes, Adam, please go ahead.
Hi there.
Hi.
So this is a super cool talk. Thank you, and I love your paper. I was very excited to hear you talk about it here. And it made me think about something that I really just don't think a whole lot about. Lauren Schumacher has sort of chided me sometimes for not thinking about noise functions across different temporal scales. I think this is the first time it became clear why it was a problem that you have to deal with in analyses. And so the thing that stresses me out about this is it just seems like it's one more layer of stuff we have to measure. Now we now have to go out and measure lots of plots. We have to measure them repeatedly through time. And now we need to have enough data so that we can parameterize some things. I mean, like even wavelet functions need quite a bit of information to fit them. So yeah. Like, do you have a suggestion for how to go about efficiently doing this in a place where you don't have 30 years of data to draw on already?
Yeah, so I think that's a great question. We confronted that a lot, and a couple of responses. The first is that you need the tool to fit your question. And so I think there's times where the classic variance ratio is fine. I stand by a 2014 paper. I think that's a cool pattern, and I think that was the tool for the job, right? And so you can pull out a lot of the kind of stronger dynamics at a system with coarser tools. And there's times where that is the right tool for the question. And so when you're doing that, then just, the lessons from this and these finer scale ones is maybe think about the fact that this is likely biased by long-term drivers and what that means in terms of your interpretation. But you could still use the tool and then say this, think about it as something that we know in terms of drivers. The second is that, one of the reasons that we ended up doing the kind of spectral approach for decomposing the variance ratio, part of the genesis of this was that the LTRs were started in the 80s, and now it was in the late teens, and we're like, there's finally enough data to do a wavelet mean field at last, and then it was like, man, this is still pretty hard. It's still, we're just enough data. But some of the spectral approaches work really well with 10 years of data. And so I would feel pretty comfortable applying the timescale specific variance ratio to 10 years of data or longer, whereas I wouldn't feel comfortable doing a wavelet mean field with 10 years. And so we tried to make tools that were a little bit adaptable, and you can't pull quite as much information from the TSVR as you can a wavelet mean field, but you still can get a lot more than from not looking at the signature at all. And then finally, I would just say this is another plug for why keeping investing in these long-term sites is so important, right? And like, it kind of sucks, wait for it, but it is a little strange. I mean, there's some experiments that I started in grad school, and I'm like, holy shit, this is a decade-old experiment now. Like it actually happens, and investing in the infrastructure and then taking things on early on and finding a way to integrate them in your life can ultimately kind of get you there in a way that happens scarily faster than you would like to acknowledge.
Cool. Thank you very much.
Yeah.
Thanks. Are there any other questions? Oh, yes, Paul, please go ahead.
Hi, Dr. Hallett. That was very interesting. I was just visiting with a friend of mine, Dan Royman, who's at the University of Kansas, who's doing a lot of work on synchrony. And one thing he's emphasized is not just looking at like covariance approaches, but really paying attention to extrema, because sometimes the correlations are in terms of extreme values, and then the rest is just sort of noise. And he's developed a series of techniques that'd be very interesting to apply to the data sets you've talked about, copulas and so forth, that deal with that. So I just thought I'd mention that.
That's a great plug. So Dan, Katie Suding and I are the co-PIs on this full working group. So,
okay.
So his fingers are all over it. So here he is here. If you check out the paper with Lauren Shoemaker, there's one vignette that I didn't talk about, which is how these patterns change in changing, like, non-stationary environments. And so we give an example of thinking about a climate shifting, and so you might have had a period of time where you have two species that are responding to a climate driver, and one of them just really tracks the driver and the other one only tracks the extremes, right? And so what you get when you have that is you can have basically this, species one is track, and species two is like, there was the extreme, there was that extreme. And so that can create a synchrony at long time scales because they're responding to the drivers in different ways. And that can shift as, let's say that's a temperature threshold, but then everything's getting warmer. You then can start to see an increase in synchrony among the species at multiple time scales, because you're crossing that threshold of the less sensitive species more often. So that's one way that we've been thinking about this extrema. And then the copula approach, and I think Shyamalan is here, and so she could pass it over to her to talk more, but I think has been really interesting in terms of what their argument is. When you're thinking about covariance, there can be tail dependencies, and so if you just do a correlation or covariance across some variables, you could get the same relationship, even with maybe it's being driven by, at the low end there's a very strong relationship and then more spread at high, or conversely. And me as the field ecologist, I think about that, like maybe there's a drought, like a really strong extreme drought, and so when that happens, you get a strong tail dependence at with a dry variable, because everybody crashes, right? And so those points of correlating two species are going to be very low with the drought, versus when it's wet, maybe there's more heterogeneity in what microsite you landed in, or competition, and so you don't have a stronger correlation at the high end of the gradient. And so Shyamalan has done some really cool work, kind of with Dan, of course, pulling that apart. And so this group, she joined our team kind of as an honorary member as the working group was finishing up. But yeah, and I think your hand is up now, Shyamalan.
Yeah, I mean, if we remember, like we also tried to see these extreme effects of gopher and other invasive species. Remember for this JASPER ridge community and tail dependence. Yeah, so yeah, we got some cool results, but ultimately we don't have time to write a paper.
Yeah, it's true. But there was some kind of where it seemed climate associated, and some where it was like,
yeah, that rainfall variability you also said, and also for some species we saw some result based on how strong their root is, right?
Yeah.
On the shallow soil or very deep soil.
yeah. And then finally, in Jasper Ridge, there's been this invasion pattern of annual grasses coming in, and there's some species that just cannot handle them. And so you got a tail dependence where a patch where the annual grass had very successfully established, we saw strong tail dependence where those other species would crash out too. And so it was an interesting metric that maybe we should pick back up, Shai and Melina, for this system.
Thank you.
Mm-hmm.
Thank you so much. Are there any other questions? While people are thinking, I might have a question. I don't know the details of how you did your spectral methods, so apologies if the question is completely off, but you must make some sort of choice about what kind of basis functions you used to do the spectral analysis. And then does it matter what you do? I assume it would, but I wouldn't know. And is there a clever way of choosing the basis functions?
This might be out of my wheelhouse. We get this nice working group coming together, and I know that Lei put a lot of thought into it, and the supplement for that paper applies it to different synchrony metrics and thinks it through. But I can't remember off the top of my head. I know that one decision point that has plagued us is what do we say is a short time scale versus a long time scale? Because when you apply the spectral decomposition, you can see patterns across any level of frequency, but then to really interpret, you want to dice and splice short and long, just so you can aggregate a little bit. And we typically do 4 years as kind of 4 and under as a short. But there's been debate about why, is that arbitrary? Is that not? Is it, so that I think was a big discussion, but I think some of the other methodology was also thought about more, but it's not my wheelhouse. So I was informing more on the ecology side of what are the decisions here.
No worries. I'll check out the supplement.
Empirical side. Yeah, check out the paper though.
I'll do so. Thanks, Lauren.
Yeah.
Are there any other questions? Yes, Andrea, please go ahead.
Hello, thanks, Dr. Hallett, for the talk. I was wondering if you had prescribed fires in any of your study sites, and what sort of responses that resulted in terms of synchrony and asynchrony post what I would perceive a more intense disturbance compared to a gopher. Yeah,
that's a great question, and intense, and also much more, wider spatial extent, right? So that dispersal patterns can be changing at these sites. So yes and no. It occurs at the sites, but not in our data set. So we, for the kind of cross site, we focused on using control plots from a lot of longer experiments. And so that was just kind of standardized, right? So some had nutrients, some had, water, some had fire. And so the Kanza prairie is burned on a fairly regular basis. And I think we made the choice to use an annual burn because that was consistent, versus if you tried to do like a 20-year, you get this, you get the phenomenon you're expecting, where it's going to be this very big effect. And then JASPER and the serpentine, pretty rare for it to burn. It's so low productivity. So it was not a place where you would do a prescribed fire for management. And we've had some natural fires, but everyone's like, oh my God, the serpentine's on fire. Let's go study it, because it's pretty rare. That said, I think that some of these sites are very amenable to testing that question. So I think Kanza would be the most obvious, because they've got these different watersheds with different burn histories. And so you could apply this from like their annual burn up to the 20-year burn, and look at that type of signature, and what is, the 20-year burn, how long is do you end up with a really strong time scale, or does it start to kind of disintegrate and then revive? And, like basically, do you get the same time scale specificity of like a 10-year burn and a 20-year burn or not? I haven't done it, but it could be really cool.
Really interesting. Thank you.
Yeah.
Thank you so much. Any other questions? If there are no more questions, then thank you so much again, Lauren, for coming and for giving that talk.
Yeah, thank you.
And I will take a look at the supplement there for sure. And so
sorry to punt to a supplement. Like I could give you a pithy answer, or I could make you read a lot, but I have to make you read. I'm sorry.
No, it's all, of course, it's all fine. So thank you again for the talk, and thank you everybody for coming. In 2 weeks, as usual, we shall resume. So we all hope to see you there and then. So thank you again for coming, and we will see you next time. Bye-bye.